Hello, everyone, and welcome to the sixth episode of our series highlighting current and exciting research projects in the MGNet Research Consortium. My name is Olivia, and I'm the program manager at MGNet, and I will be your host for today. I am joined by Dr. Fatemeh, who is a postdoctoral associate in the Department of Neurology at the Yale School of Medicine. She has a research background in autoimmunity, and she was chosen as this year's MGNet Scholar. I'm also joined by Miss Melissa Wolhust, a patient advocate for the MGNet External Advisory Committee and a longtime MG patient herself. In this episode, we will feature Dr. Khani's project, B-cell depletion and refractory response to anti-CD19 therapy in myasthenia gravis. I will now give you both the floor to discuss this important topic in MG.
Thank you so much, Olivia. Dr. Khani, first, congratulations on your 2026 MGNet Scholar Award. It's quite an achievement. And as I was going through your research and some of your background information, I was really struck by your commitment to research in MG. And I was wondering if you can tell us a little bit more what brought you to that line of research of myasthenia gravis and what keeps you committed to patients that are dealing with myasthenia gravis?
Thank you, both of you, for that nice introduction. And it's truly an honor for me to be here with you today. So what drive me to study myasthenia gravis is, in many ways, MG is a paradigm for autoimmune disease, especially the ones that are antibody mediated. So when we study myasthenia gravis, that not only help patients with myasthenia gravis, but that also will benefit other patients suffered by other type of autoantibody-mediated disease. But what keeps me committed to study myasthenia gravis goes beyond the science itself. It's actually in early career, when I just started my position here as a postdoc, I received an award from MGFA. That was a 3-year award to support my research. And that was a generous donation by Danny McSpadden in memory of his wife, Jackie McSpadden, who had MG herself. And that fellowship didn't just support my research, but that also introduced me to patient community and helped me to see the disease from the eyes of people who are living with it. So that feels like a commitment to meet people and meet Jackie's family. And later, just recently, I received the MGNet Scholar Award. That feels like the continuation of the same commitment to study MG.
Thank you so much for giving us that insight. So often we as patients, we just see kind of researchers up here in their lab work, and we don't understand the commitment that you guys are making to study our disease and also the science behind it and how it can impact the lives of others. So I truly appreciate that insight. And in terms of your project, what's the central question that you're attempting to answer with your project?
That's a really good question. During my postdoctoral research, I focused on response to therapeutic options in myasthenia gravis. So it's basically what I did was study what separates patients who respond to a given therapy versus the one who does not show the clinical improvement, and we call them non-responders. So first of all, to study the response mechanism, we first need to know the pathology of the disease. And as you know, MG is mediated by antibodies. The antibodies in myasthenia gravis, they target neuromuscular junction. If it's a neuron, this is a muscle cells. This is that space is the neuromuscular junction, and the antibody in myasthenia gravis binds to muscle cell surface and interrupt the signal transmission from neuron to muscle, and that leads to fatigue and muscle weakness. And antibodies are expressed by B cells. B cells are a part of the immune system, so they're essential in human bodies. But in the autoimmune disease, a subset of B cells, they start to produce antibodies that by mistake recognize different part of body as pathogen. So it should not happen, but in autoimmune disease, that happens by mistake. And that antibodies in MG detects two known antigen on the muscle cells called AChR or MOS. So that's how MG pathology happens. So as I mentioned, antibodies and B cells are the core in MG pathology based on what we know today. One of the therapeutic options that just got approved by FDA is the B-cell depletion therapy. It was approved in December last year, and the mechanism of action of this drug is if this is the B cells, that drug is an antibody. It binds to the cell surface of B cells. It recognizes the molecule that is specific for B cells. So upon binding to the B cell, that induces the B cell death. So it lowers the B cells count. And as a consequence, because B cells express antibodies, lowering the B cells would lower the antibody levels, right? So in theory, patients should respond because those pathogenic antibodies will be decreased. In a recent clinical trial that published last year, they showed that the majority of patients, AChR and MOS patients, showed clinical improvements when they received the treatment. However, about 30% of patients, they didn't show clinical improvement. And this is the core question in this project, that what separates patients that showed improvement versus the one didn't benefit from the treatment. So in this project, I'm trying to find a biomarker that can distinguish responders from non-responders. And the non-responders are the term we use for patients who do not respond as well as other patients do.
Okay, to the B cell depletion. And thank you for explaining the role of B cells in the disease process also, because I think as patients, I don't historically we hadn't heard a lot about that in detail, and it's really helpful to just understand the whole process. So in terms of your research, why does it matter for patients living with the MG, like from day to day? What could change because of the work that you're doing?
Yeah, so as you know, comparing 2 years ago, there are more therapeutic options available for patients with myasthenia gravis. And there are many more in pipeline and many in the clinical trials. So in the near future, there will be even more therapeutic options available to treat myasthenia gravis. And that's a real progress. That's great. But at the same time, it raised the critical question that what's the best treatment for a given patient? Because for now, what happens is physician prescribes medication for patients and see how patients respond. And then if it works, great. If it doesn't, they change the therapy and see how patients responds to the treatment. So it would be a burden on patients to go through different rounds of treatments. So this project will help, we hope, to find a clinical biomarker that will distinguish at the beginning which patient would respond to B-cell depletion therapy. So physician would have informed choice when they, decide which medication to prescribe for patients.
Yeah, I can say especially as a seronegative patient, so, sometimes we'll say we're kind of like guinea pigs, we're just like, well, we'll try this, we'll try that. And thankfully there are some excellent neuromuscular pet specialists out there that are pretty great at honing in. But this research really sounds like it's groundbreaking in terms of being able to target better treatments. In speaking about being seronegative and antibodies, I know that your research is specifically in the AChR area. However, it was interesting to me that I saw the acknowledgement that a lot of us as patients experience the serum antibody level is not necessarily correlating with the severity of the disease. So someone can have high antibodies and be functioning almost normally, and somebody like me is below the baseline level, and yet I've been in crisis and had the whole spectrum. So this shift in looking at what the antibodies are actually doing versus the number of antibodies. What does this shift mean in terms of really your research and beyond? It seems like a big shift.
Yeah, it is. That's an excellent question. So the mismatch that you're describing is something that we work on, and we published a paper recently. So it was about the mechanism of response and in a type of treatment called FcRn inhibition. So the FcRn is responsible to recycle IgG. That's the part of what the human body does. It recycle antibodies to keep the antibody level high as much as possible, because antibodies in theory are good, but in autoimmune disease they start to target self body. So the treatment, the FcRn blocker, they inhibit the recycling of antibodies. So it will decrease the antibody level. So in theory, everyone should respond, because in every patient treated with the medication, the antibody level dropped. However, about 30% of patients didn't show clinical improvement, and that was, surprising. So we studied that cohort of patients, and we understand that although the patients that didn't show clinical improvement had a reduction in the total antibody level, the remaining antibody were enough to induce pathology. So it's not about the quantity of antibody, it's about the quality of the antibody. So if a patient has antibodies that are more pathogenic, even at lower level, there is still enough to induce the disease. So it shows us that for that subset of patients, that for those non-responders, we need another type of treatment to help patients to feel better. And that really matters for patients, because research like this, they pave the way to personalized medicine. It means we tailor treatment for each patient needs, and that stop patients from going through different rounds of treatment.
Yeah, this is incredible. The line of research and how far out it can spread into helping those of us patients with with autoimmune issues and antibody issues. So when we're thinking about your project and the impact moving forward, the short-term impact, and then further down the road, the long-term impact. Can you tell me what your hopes are in terms of for the patients, where this will go?
Yeah, so for a short term, this project would help us to understand the science behind patient response to B-cell depletion better. And hopefully that will help us to identify a clinical biomarker so that would help physician to be able to have informed choice to prescribe B-cell depletion for patients or not, or decide to prescribe another type of treatment because B-cell depletion may not be helpful for the given patients. Farther ahead, the approaches we are developing in this project would also help to study the response, the mechanism of response to other treatments that will be available for myasthenia gravis. And also more broadly, it will help other patients suffering from other type of antibody-mediated disease.
This is wonderful, and I'm so honored that we've been able to have this discussion today and for you to speak on where research is going in terms of myasthenia gravis and even beyond that. And I have so much gratitude for the work that you're doing, the passion that you have, and dedication to that work. And I truly appreciate the time that you've taken today. Thank you so much for your time, Dr. Hadi.
Thank you, Melissa. It's just an honor to me to be selected as the scholar this year, and also to be here to have this interview with you. And it's deeply rewarding for me to know that this research will benefit patients' health and well-being, and that's really what keeps us motivated and committed to the field. Yeah, thank you.
Thank you.
Yeah, thank you both so much for participating in this conversation. I think these discussions are super important and helpful for patients with MG and their families. We hope that this conversation will be a great resource for the MG community and will give more insight into what is going on in the world of MG research and what to look forward to. I thank you both for your time.
Thank you, Olivia. Thanks, Dr.
Thank you.
Thank you.